I'm here to talk about my thesis, which was an investigation into the impact of ocean acidification on coralline algae. So, firstly, some background information on coralline algae. They are red macroalgae, which belong to the Corallinales order, and they're characterized by having a hard skeleton formed by the deposition of calcium carbonate into their cell walls. They can be split into two groups based on growth form, and these are genicular, also known as articulated, and non-genicular, also known as crustose. I think it helps to think of them by their alternate names because it gives a bit more of an indication about what the algae look like. So the articulated algae, which you can see in the top picture, have flexible branching fronds, which are made up of calcified segments separated by uncalcified regions, whereas crustose coralline algae, seen in the bottom picture, are fully calcified, and they can grow as a crust over other substrates or as free-living organisms. The two species seen in these pictures are the adult species I used in my study. So on the top, this is a Jarnia species of the articulated, and the bottom is the crustose species, which was a Phymatolithon species. Due to the lack of detailed understanding and uncertainty in species designations, these were only identified down to the genus level for this study. So coralline algae in general have a widespread distribution. They can be found from the intertidal zone all the way down to depths of 270 meters, and they're present globally from the tropics to the poles. They perform a range of different ecosystem functions, and they have a very complex life cycle. They can reproduce sexually or asexually, and there are a number of different spore stages, some of which are also self-perpetuating. So the spores in the external environment are not calcified, and calcification only begins after they have settled onto a substrate. So in my study, I've defined recruits as newly settled algae, which are able to be seen attached to the substrate of the tanks. Now, some background on ocean acidification. As is widely known, since the onset of the Industrial Revolution, 30% of the carbon emissions released have been absorbed by the oceans, which has led to changes in the seawater chemistry, including an increase in acidity. The pH of the oceans, the average pH, has already decreased by 0.1 units, and this has happened because when the carbon dioxide dissolves, it quickly reacts with water to form carbonic acid, which then breaks down into bicarbonate and carbonate and releases hydrogen ions. By 2100, the average decrease is expected to reach between 0.3 and 0.5 units. It's thought this will have a large impact on coralline algae, as their skeletons are composed of a high magnesium calcium carbonate polymorph. This is the most soluble form of calcium carbonate. So in this diagram, you can see simplified representations of processes which are vital to coralline algae. In this box is calcification, which combines either bicarbonate or carbonate with calcium ions to form the calcium carbonate in the skeleton of the algae. And then along the top here are the processes of photosynthesis and respiration. So an important point to remember later for the methods is that the processes of photosynthesis and respiration do not change the buffering capacity of the seawater, whereas calcification does due to the deposition of bicarbonate or carbonate in the skeleton in the form of calcium carbonate. And the buffering capacity of the seawater is its ability to neutralize hydrogen ions. The aims of my study were to determine the effect of ocean acidification on the metabolic and calcification rates of two different species of coralline algae across 4 different pH treatments. For these two species, I aim to measure photosynthesis, respiration, and calcification of adult specimens. And then additionally, I aim to measure the recruitment of coralline algae in general, as I could not determine which species each of the recruits belong to. So my hypothesis for the photosynthetic rates was that they would not be significantly affected by pH, reasoning that the process of photosynthesis in itself raises the internal pH through the removal of carbon dioxide. For respiration, I hypothesized the rates would increase as pH decreased, as the energy requirements needed to maintain normal cellular conditions would increase. For calcification, I thought that the rates would decrease as pH decreased, as the deposition of calcium carbonate is less chemically favored at low pH. And then finally for recruitment, I thought a similar thing in that the recruitment would decrease as pH decreased because it becomes more difficult for the algae to form their skeleton. So my experiment was set up in a way to mimic the natural conditions of that algae species I've used. So they're from coastal environments, and in these coastal environments, there is a daily cycle of pH where due to the photosynthetic activities of marine life in the light, the pH decreases, and then at night when it's dark, it drops again as only respiration is occurring. So in all of my treatments, I have this cycle throughout the course of the day, and here is the ambient treatment, where the average across the day is 8.03. So in this graph is all my 4 pH treatments, which decrease from ambient 8.03 to 7.93, 7.83, and then 7.63. And again, this is the average of the treatments over the course of the day. As well as this pH cycle, there was a daily light cycle, and the spectrum of light emitted onto the algae was chosen to have high blue and green wavelengths, which is what they experience in their natural habitat. For the setup of the tanks which house the organisms, they were spread across 8 of these water baths, which are fed water from these white header tanks. There were 2 header tanks for each of the 4 pH treatments, and the water in them was pumped in from the sea outside the lab and then modified directly within each header. If the pH needed decreasing, CO2 was bubbled into the water, and if it needed increasing, air was bubbled in. So the algae I used were kept in these experimental conditions for 76 days. After the 76 days were up, I performed incubations on the algae. So I had 48 specimens in total on which I performed incubations, and these were comprised of 6 replicates in each of the 4 pH treatments for both the species. And for each specimen, there was an incubation carried out in the light and one in the dark. So in terms of what an incubation involves, the algae is placed into a chamber, like you can see here, which is completely filled with a known volume of seawater. So before the incubation, the pH and dissolved oxygen of this water are measured, as well as a sample taken to later calculate total alkalinity. I'll explain what that is in just a second. And then the chambers are placed on a mixing plate in this water bath and left for an hour and a half. And then once the hour time is up, the same pH and oxygen measurements are taken, and another sample for TA, and then the difference between the start and the end of the incubation, you can use that change to work out what the rates I looked at are. So total alkalinity is the ability of the water to neutralize acids, i.e. the buffering capacity of water, which I mentioned earlier is not affected by photosynthesis or respiration, but does change according to calcification. So you calculate total alkalinity by taking the collected samples and performing an acid titration. And the data from this titration is put into the R package C-carb, which gives a value, and then the change in total alkalinity over the incubation can be used to calculate the calcification rate. So after I'd finished the incubations and the experimental setup was deconstructed, I took each of the experimental tanks. They were cut into 5 pieces, so 4 sides and a bottom, and then I took photographs of each of these pieces. Using the image analysis software IMBRSea, I then took each photograph. It was adjusted so that only the recruits remained, and then using the analyze particles function, you get the area of each recruit. This area was then summed for the whole tank and compared between treatment groups. So now onto some results, but first I will mention some statistics. So when I'm comparing rates between pH groups, either a one-way ANOVA, a Welch's one-way ANOVA, or a Kruskal-Wallis test was used, dependent on if the data was normal and if the variances between the groups were homogeneous. So for the photosynthesis rates, I found no significant differences between the treatment groups in either of the species. And you can see on the graph, the Jarnia species is in purple and the Phymatolithon species is in grey. So this agreed with my hypothesis of no difference between groups, and was also consistent with the literature, as other studies have found little effect of pH on photosynthetic rates. Work on coralline algae and other marine photosynthetic organisms has suggested that in the future, changes in light and temperature will have much more of an effect than pH on coralline algae, as these factors they cannot regulate themselves, whereas algae have some control over their internal pH due to the process of photosynthesis. For respiration, I hypothesized that the rates would be higher at lower pH as stress increased. However, I again found no differences between the treatment groups for either of the species. So the ability of these organisms to cope with the different pH levels could be due to the fact that they come from coastal environments which experience fairly large changes in pH daily, so they may have adapted to cope in a range of pH conditions. They also could be regulating their own pH, so the pH of the bulk seawater doesn't have much of an effect. They could be doing this through the regulation in the diffusion boundary layer, or the DBL, which is largely done through photosynthesis. And it's also been shown that interactions with other species can have an effect. For example, interactions with turf algae have been shown to increase the pH in the DBL, and the microbiota associated with the coralline algae have been shown to provide some level of protection against acidified conditions. So if we then look at the calcification rates for the Jarnia species, I found no significant differences between the treatment groups in the light or the dark, and the light is in the light blue and the dark is in the dark blue. So it was expected that calcification should be higher in the light and lower in the dark as the pH is higher in the day. However, this was not found, and the rates were close to 0 or below 0 for all of the pH treatments. Whereas for the Phymatolithon species, the trend followed as expected in that the calcification rates were higher in the light and lower in the dark. Although there were no significant differences between the treatment groups in the light or the dark, I do think there's a slight trend in the dark that calcification rates are higher at the higher pH. This is because in this 8.03 treatment, 5 out of 6 of the replicates had positive calcification rates, so they were actively calcifying, whereas in the lowest 7.63 treatment, 5 out of 6 of the replicates had negative calcification rates, which indicates dissolution. So if we now look at net calcification, which is the calcification in the light and the dark combined, and compare between the two species, again with Jarnia in purple and Phymatolithon in grey, you can see there are differences. So there are significant differences between all of the groups except the 7.93 treatment, when I compared using either a Welch 2-sample t-test or a Wilcoxon rank sum test. And these differences were that the Phymatolithon rates were higher than the Jarnia rates. So as you can see on the graph, many of the Jarnia organisms had net rates below 0, which I've marked with this red line. And this was happening even at ambient conditions. So as this was happening across all the pH treatments, it suggests a factor alternative to pH was causing stress. One factor could be that the specimens for the Jarnia individuals were attached to bases at the start of the study to anchor them down. However, over the course of the 76 days, many of them became unattached from their bases, so they were free to move around the tanks, which subjected them to physical damage. The Jarnia species in general is more delicate, so is perhaps less suited to lab studies than the crustose Phymatolithon species, which was actively growing across the board. Now onto the results of recruitment. I compared between the pH treatments using a Kruskal-Wallis test and then did pairwise comparisons using a post-hoc Dunn test. And I found that the average total area was lower in the 7.63 treatment than the 2 intermediate treatments significantly, so it's significantly lower. So this could be due to some level of inhibition of recruitment at this low pH. And a 2013 study found that the amount of recruits was reduced in acidified conditions due to more abnormalities and a higher rate of mortality. And then a biomechanical study from 2018 showed that the number of recruits was reduced due to a reduction in attachment strength at low pH and initial attachment strength, and then attachment strength over the course of the time. So in general, because of my methods, I cannot tell which factors have caused this reduction in total area at the low pH. However, it's not surprising overall that the biggest effect of pH I found on coralline algae was in the recruits, which are the early life stages, as these early life stages are more susceptible to stress, whereas my incubations were performed on adults. Coralline algae are generally long-lived organisms, and some have lifespans of hundreds of years, so it's much more likely they will be resistant to changes. It's also been shown that over multiple generations, coralline algae can gain tolerance to ocean acidification, so perhaps looking at just one generation of adults is quite limiting. So to conclude, I found no effect of ocean acidification on the photosynthesis or respiration of either of the two species I studied. The Jarnia species failed to behave as expected, even at ambient pH, suggesting there was an effect of lab conditions. The Phymatolithon species showed a slight trend of lower calcification rates at low pH in the dark, however, this was not significant. The recruitment of coralline algae in general was significantly lower at the lowest pH treatment. And finally, future studies should investigate the effect of ocean acidification across the whole life cycle of an individual, not just those collected as adults, and across multiple generations. A very big thank you to Dr. Cornwall, Eric and Luya for having me in their research group and for all their help and guidance. Thank you very much for listening, and please let me know if you have any questions.